Okay, so today I'm going to be talking about antigenic variation. Typically, I'm going to be talking about antigenic masking and antigenic mimicry. This is different from antigenic drift and antigenic shift, which I had talked about in a previous video. Antigenic drift was what? That was when the surface glycoproteins on a pathogen, specifically RNA viruses, changed over time gradually due to mutations because RNA polymerase lacked proofreading. So these mutations occurred, the antigens on the pathogen changed, and it made it harder for antibodies to detect or your immune system to detect these pathogens. Antigenic shift, however, was when it was essentially recombination or reassortment, genetic reassortment of two different pathogens, and it created an immediate shift in the surface glycoproteins of a pathogen. So obviously, we talked about that antigenic masking and antigenic mimicry is of course different. Antigenic masking is essentially when the pathogen will use host molecules to hide from your immune system. So usually these are done through proteins. So they'll take plasma proteins. Plasma proteins are proteins found in your blood, and they will kind of cover themselves with them, attach it to themselves, and it'll act as kind of like a camouflage, right? I wrote it down actually. They will sometimes use the host cell's membrane, similar to how like when they would lyse out, they would take some of the membrane with them. They could also use glycoproteins or antibodies. Antigenic masking is essentially they are putting on a mask, they are putting on camouflage to hide from your immune system. This makes them very hard to detect, obviously. That's why it's so useful for pathogens and bacteria, etc. So that's antigenic masking. Now, antigenic mimicry is a little bit more complex, I suppose. Antigenic mimicry is essentially when the pathogen will express or produce their own antigens that will look like the host's own molecules. So instead of taking the host molecules, they will produce and imitate molecules that they find within the host. That way they can hide from your immune system. So instead of hiding, it essentially imitates the host cell. This is pretty negative. Of course, this is really negative for us just because sometimes this will cause autoimmune responses, autoimmune reactions within our body. Now, what I mean by that is if, say, you have a bacteria, I'll get into an example in a second, but say you have a bacteria that is producing its own antigens that mimic something else found in your body, and your immune system detects this. Your immune system says, okay, this is obviously a pathogen we don't want in our body. It starts targeting that bacteria. The downside is that because they have these antigens that mimic something else in your body, your immune system will not only target the bacteria, but it'll also target whatever that bacteria's antigens are mimicking, if that makes sense. So it'll cause like an autoimmune reaction within your body. A popular example of this is Streptococcus pyogenes, which will essentially produce M proteins that look like heart tissue, human heart tissue. The problem with this is that your immune system will make antibodies against Streptococcus pyogenes, but it'll also cause antibodies that will cross-react with the heart tissue in your body, causing rheumatic fever. So there is some negative, obviously there's negative effects with antigenic masking too, but the biggest thing with antigenic mimicry is it'll cause these autoimmune reactions within your body. But yeah, so antigenic masking is essentially camouflage by taking the host molecules within your body and kind of attaching it to their surface. And then antigenic mimicry is when they express and produce their own antigens that mimic other cells or other molecules that your host body produces. Hope this was super simple. Hope I made this quick and easy. Yeah.